Also very interesting. So maybe we can go ahead and get started. Thanks for being here. Thanks for being at this session in proteases and proteins. So my name is AJ Vinchelli. I'm about halfway through my PhD program at the University of Massachusetts, Dartmouth. I'm doing my capstone project with Faraz Khatib. Faraz is a folded guy, and he's usually at RosettaCon. He's not able to make it this year, so I'm here in his stead, and I'm very glad and honored. I'm here today to introduce our two speakers for this afternoon's session. The first of which is Dr. Chris Ball, who probably doesn't need too much of an introduction, but he is the head of protein design at the Institute for Protein Innovation and Harvard Medical School in Boston. Chris received his PhD in molecular and cellular biology from Dartmouth College in New Hampshire, where he studied the molecular mechanisms of bacterial host-pathogen interactions. Chris performed his postdoc research in the Baker Lab at the University of Washington, developing computational design methods and laboratory tools to create scaffold peptides de novo. Today, Chris is going to be talking to us about the discovery and engineering of enhanced SUMO proteases. And please join me in welcoming Chris.
All right. Well, thank you for the introduction, AJ, and also welcome AJ to the Rosetta community. I think this is your first RosettaCon, so it's great to have you. So today I'll tell you a story that really was a side project that I started in the Baker Lab with a team of really talented undergrads and finished after I left. And it was a lot of fun, and it's published. So again, if you want to go check out Frank's stuff, you won't offend me. So I was thinking a lot about in the Baker Lab how to make proteins and how to make sure that the proteins that I'm testing I get the most information out of. And I think something that everyone runs into is you express a bunch of design proteins, you worked really hard, and you want to know, is this going to work? And some stuff you just can't make, and you never know if my design was bad or if there was something with the bug or the gene, and who knows why those proteins don't purify. And so if you use SUMO fusions, so this is a ubiquitin-like protein, small ubiquitin-like modifier. If you fuse proteins to this and you express them, you can make most things. And I found that I can make designs that are unfolded and just bad, and I can purify them, and I can test them, and I can see that my design is really bad and actually get a true negative. So I recommend SUMO fusions if you're not using them. They're really great. And the other really cool thing about SUMO fusions is that there's a protease that recognizes the folded SUMO protein and then specifically cleaves it. And it does so without leaving scars on your protein. And you can stick stuff on the other side of the SUMO protein, like a his tag, to facilitate protein purification. The enzyme that does this is called ULP1. It's from Saccharomyces cerevisiae. It's a bigger protein. There's two domains. One is a protease domain, one's a regulatory domain. And the reagent enzyme that we use, you clip off the regulatory domain. So you just have this protease domain. And while it's a really nice specific protease, it is also a little bit finicky because it's half a protein that was from, some natural source. So in my hands, it was not super stable. It couldn't really get it to concentrations above 2 mg per mil. So when you're trying to prep a large batch as a reagent enzyme in the lab, you have solubility issues where it's just crashing when you elute it from the column. And if you buy the commercial stuff, it tells you have to put detergent in your buffers, which is also not really ideal for lots of things. And a lot of times the digests that I was doing weren't running to completion. So you lose a lot of yield that way. So it was just a pain. So I wanted to fix this and maybe learn a little bit about why this was happening along the way. So if you think about, okay, so my protein is not super stable or super soluble. The solution properties are kind of bad. So I want to fix this. How do you do that? Well, you can take the enzyme itself and try to make it better. Or you can try to find an enzyme that might have the same function from a different organism that might just be better. And one of the ways to do that is just look in thermophilic organisms, right? Most reagent enzymes are proteins from thermophiles. So the first approach, just looking at the enzyme that we were currently using, here's a co-crystal structure of the SUMO protease, so it's in color here. Sorry, let me not... there we go, so people on both sides can see. So it's in color, and then the SUMO protein is in gray here. And you stick this on the N-terminus of whatever protein you're trying to purify, and then this is the little connector where you stick your design protein. The SUMO protease recognizes this whole SUMO domain and clips it off. So the residues that are involved in the direct recognition of SUMO are in yellow. Active site residues are in green. Then the rest of the protein is really cyan, the polar residues. The orange spots are solvent-exposed residues that are just sticking out. They're probably important for whatever this protease is doing in Saccharomyces, but if you want a reagent enzyme, these hydrophobic surface residues are just presumably problematic. So this seems like a fairly straightforward design problem. So just to briefly go over how this works, record the starting energy, the FA sol, then do a flexible backbone design, but you don't want the protein to move too far, and then you just check to make sure that your protein actually didn't wander too much during your design. So to go into a little bit more detail about this, because this is RosettaCon, there's been I think a really big transition in the Rosetta field in the last really 5 years. When I joined the community, every design project was really like a piece of handcrafted art. You had some protein and you had an expert who would look at a model and they would write a protocol specifically to do that design. Often there was a res file that was custom made for that individual molecule. And it worked, but it wasn't very reproducible. If you wanted to do the design again, that same task on a different protein, you had to do all of the manual work over again. And really the inclusion of residue selectors has kind of broken this open. And so now you can write reusable protocols in Rosetta scripts that you can just turn a crank and do this rather sophisticated design method without needing any human intuition or skill or talent. You can put a black box around this complicated method and make it so that other people can access it. And so just really simple logic, right? I mean, we want residues that are on the surface, and we want hydrophobic residues. And we want to design those, and we don't want to design everything that's not those. And now you have a way to pick out these problematic residues on any protein structure. I think also the original mindset is keeping the backbone fixed, but our protein models have some measure of error. If people were in the last section listening to Jelly's talk, right? Crystallography is not a perfect art, so there is error in these models, and allowing the backbone to move within the expected error of the crystal structure, I think, is really important and improves the accuracy of your designs. And so in this case, I just allow the backbone to move by about 10% of a carbon-carbon bond. And don't forget to turn it on in your energy function. And I brought this point up a couple years ago at Winter RosettaCon, and I just want to highlight it again. There's this, I think, permeating idea in the Rosetta community that there's common knowledge, but sometimes the common knowledge isn't as common as maybe everyone might think it is. And so there are certain task operations that if you're doing design, you pretty much always want these, and they're very important. And there may be more of them that I'm not aware of that maybe are common knowledge in other labs. And we have a place to collect them on the Rosetta documentation. So if you have some task operations that you think should always be used, please include it. And so, for instance, like this is limit aroma chi. So there are certain rotamers of aromatic amino acids that only appear in really unique structural motifs. And unless you're designing that specific motif, you probably don't want to sample those particular rotamers of aromatic residues because they won't form that way. And so you want to just disable sampling those during design pretty much always, including extra rotamers in the design, not throwing out the starting rotamers. And this is a sequence profiler that makes it so when you're looking at designing residues in a loop, there are certain residues that you just don't see in particular geometric arrangements in the PDB. And so it's probably a good idea to not try designing those. So anyways, common knowledge, it's good to be common. Okay, so here's just an example of what this actually looks like. So here's our polar residues, and they're now, non-polar residues, and they now become polar. So the design methods works. And there's a little bit more to the protocol, so we automatically detect the protein interface, and then the one manual component is picking out the catalytic residues and just saying, I don't want these changed. Okay. So that's a pretty straightforward way to do things in Rosetta. The other approach is to try to find a homologue from another organism that might have just better properties. So I mentioned thermophilic enzymes are great. Well, there are no hyperthermophiles in the eukaryotic domain of life. SUMO is only in eukaryotes. Prokaryotes don't have the SUMOylation system, so we can't get a hyperthermophile. The closest we can get is a thermophile. So it's this fungi Chamotomium thermophilum. It grows in compost heaps and can happily grow up to 60 Celsius. So it's pretty good. This organism is quite popular in the structural biology community because proteins from thermophiles tend to crystallize better. So a lot of really intractable proteins like nuclear pore complex proteins that are very flexible. A lot of people are using homologs from Chamotomium for this. It was just sequenced a couple years ago, and it's still rather poorly annotated. So it just took the SUMO protease ULP1 from Saccharomyces and just blasted the genome of Chamotomium. And the top hit only had 28% sequence identity over the protease domain. It's not super encouraging, right? 28% identity. It's unlikely that it's going to recognize the same exact substrate. Maybe it doesn't even have the same enzyme activity. It was annotated as protease-like protein. So, that's kind of a mixed bag. You'd expect probably to get it wrong. So, putting something into Robetta to predict the structure is a pretty low investment of energy, so I figured, well, let's see what this looks like if you were to predict the structure. And surprisingly, it was very, very similar to the Saccharomyces enzyme was the result. And the striking thing is that the catalytic machinery was all conserved structurally, and even many of the residues that interact with the SUMO protein itself were also conserved. And so that 28% identity is pretty much all at functional sites. So that was encouraging. So I figured, let's try it out. So we ordered genes for 4 of the Rosetta redesigns of the Saccharomyces enzyme, and then the 1 protein from Chamotomium here. So there's only 5 test proteins. And so the Saccharomyces enzyme, the sort of the gold standard, the starting one is ULP1 wild type here, and then the variants that we redesigned with Rosetta are the R1 through 4 series, and then the SUMO protease from Chamotomium is the CTH SUMO protease. So we try to adopt the nomenclature, like Taq polymerase is named after Thermus aquaticus. So it's trying to use a standardized way of naming things. So first off the bat, it was encouraging is we could purify all these proteins. All the Rosetta design ones also we could purify. We stuck them in the CD, and you can see that the wild type protease is this sort of nice mixed alpha beta character, and when you cook it, it fully denatures and forms a precipitate in the tube in the CD, and it melts with about a 40-degree melting temperature. Nice cooperative unfolding. What was really interesting, and I think rather unexpected, is these Rosetta redesigned variants. None of them fully unfold. So after you cook them to 95, the secondary structure flexes quite a bit, and then when you pull them out of the CD and look at the cuvette, nothing is precipitated. And you can see that the proteins aren't crashing out all the way. You're still retaining quite a bit of structural character even at 95. You get some unfolding of other secondary structure, but it doesn't fully denature. We don't really understand why. And they don't quickly renature, but if you incubate them at room temperature for a few days, they'll eventually refold and regain activity. And the thermophile has a higher melting temperature and fully denatures, and has a nice cooperative unfolding transition. And it's warmer than the Saccharomyces enzyme, as you'd expect, but it's still actually not all the way up to 60 where the organism can grow. Probably the other half of the protein is important for stability. So we measured the solubility of these proteases, which is really the thing we cared most about. And measuring protein solubility is really difficult, or I should say is a challenging thing to do, because the intuition is like, well, let's just concentrate this protein for a while and see when stuff starts to crash out. But that's actually really technically challenging. So the way, if you want a quantitative measurement of protein solubility, the way to do it is to add precipitant. And so in this case, you titrate the protease versus an increasing concentration of precipitant, and the protease concentration, or the solubility of the protease, will decrease as the amount of precipitant increases, and then you can give this a log transform fit, a best fit line, and then the y-intercept actually ends up being the maximum theoretical protein solubility. And this is a theoretical solubility, so it's, obviously solubility is dependent on many things, temperature, salt concentration, lots of things. But this allows you to do a direct comparison of the solubility between these enzymes. And so in the best case, we're able to get over a 12-fold increase in solubility just from designing 10 surface positions of the wild type Saccharomyces enzyme. And our protease from the thermophile is more soluble, but not dramatically so. The solubility roughly tracks with the amount of solvent-exposed hydrophobic surface area. So as you decrease the amount of solvent-exposed hydrophobic surface area, you increase the solubility. That's rather intuitive. But it didn't track, the differences overall were quite mild, right? So the least soluble one, the starting enzyme, is 55% of its surface is hydrophobic. This is just atoms in general, not residues. And the most soluble one here, has less than a 5% difference in total surface, but it changes its solubility a lot. And some of these ones that have really similar amount of hydrophobic surface can have very different maximum theoretical solubility. So there's clearly a lot more going on to this, probably charge-charge interactions, things like that. So this could be a really nice model system for anyone who's really excited about studying the determinants of protein solubility and trying to engineer those with more precision. I know there's been some effort in that in the Rosetta community in the past. Okay, so running out of time, so I'll try to punchline this quickly. We wanted to see if these enzymes still work. So we have a FRET assay for protease. So obviously, you stick a fluorescent FRET pair on the outer side of your SUMO substrate, and if it cuts, then FRET signal goes away. So we can use this to measure the enzyme activity, and all the Rosetta redesigns essentially have indistinguishable activity from the Saccharomyces enzyme, which is quite nice. The thermophile still actually chews up the Saccharomyces substrate, but it's with really different kinetic properties. So the Vmax is much higher. So you can see the scale of these axes are really different. So the Vmax is much higher, but the Km is also much higher. So it binds the substrate more weakly, but it processes it potentially much faster. And then just a summary table of everything. You can see I'll skip all this again because it's points I've already made. We also get a structure of the thermophilic enzyme, and it shows that the residues that bind to the substrate, the ones that we thought were conserved are conserved, and there are some really interesting differences. And it seems like the chematomium SUMO is probably different enough that there is some evolution. These interfaces have drifted. And Roberta does a pretty good job. So RMS 2 angstrom. Okay. So to wrap up, we've developed a generally applicable protocol for improving protein solubility. You can use this pretty much for your own favorite protein. Structure prediction actually could be a really useful tool for annotating function to really low identity genes where you're just not sure what this could be. And these new proteases are just better enzymes. So I know the Baker Lab has adopted these and sort of the gold standard reagent now. And I definitely encourage you to as well. We've got these available on Adgene. I recommend either using this CTH SUMO protease. It's much faster, so if you've got a big excess of substrate, you can huck it in and it cleaves it really well. If you need to cleave smaller amounts of protein or at lower concentrations, I would use the redesigned Saccharomyces enzyme because it's just able to work better with lower substrate concentrations because the tighter Km. So this was a really fun project. Give a special thanks to Kara Lau and Vlad. So this was a collaboration with Chris Lima, who's really the SUMO protease expert in the world. And Kara was an undergraduate when she was doing this. She's just started med school at UCSF. And obviously David for supporting me. And coffee kind of makes the world go round. And so I started this project at the Institute for Protein Design, where we had this beautiful artisanal machine where you pack your own grinds and make it. And I think this just kind of illustrates the philosophical evolution that I've been undergoing in this past year, moving to the Institute for Protein Innovation, where we have a equally fancy coffee machine that is fully automated. And with that, thanks for your attention. I'll take any questions. Colin.
Yeah, you mentioned the lower Km, but at the end we're tacking on to how the SUMO might be slightly different. Have you tried check the KM against the thermophilic SUMO?
Yeah, that's a great question. So if anyone couldn't hear, Colin was asking, we checked the KM with the thermophilic homologue of SUMO. I made the construct and tested it. I showed it was activity, but that SUMO is also a slightly bigger protein. When you clip off just that useful SUMO domain from the thermophile, it wasn't super stable, or I didn't think it was going to be. It wasn't a beautifully behaved protein, put it that way. And so I didn't think it would be very useful as a fusion protein for making protein. And so I just canned it because I didn't think it was going to be useful as a tool. But I probably should have actually tested it to see what the biological function was.
What about reverting some of the mutations that engage with the
SUMO? To try to make the thermophile have a better KM, so it's a much better enzyme. Yeah, we thought about that. There are a couple structural features that are a little bit different. We probably should have gone through and done that. Honestly, it was kind of like at the point of like, well, it's good enough. It fixed the problem I was originally going after, and the goal of, I guess, making the enzyme better would be so you could use less of it. But the prep is really crazy. You can get like 1/4 of a gram of protein out of a 1/2 liter culture. So it's a problem you can solve by just hucking more enzyme at it. Yeah.
Maybe one last question?
Oh, Amanda?
So the cross-stabilization server is an existing Rosetta tool to supposed to be generalizable to stabilize protein.
Right.
Did you try running your protein through that and comparing the sequence results?
I didn't. So PROS is really great, but that's really for trying to stabilize a protein that's unstable, right? So you're trying to make it more stable. This protein's already really stable. It's got the melting temperature is fine. It's got a great cooperative and folding transition. It's a stable protein. The problem is that it's not soluble. And you can be insoluble because you're unstable, or you can be insoluble because the surface properties are bad. So we didn't try PROS, but that is a great idea.
Great. Let's thank Chris once again.
Cool.